Hi, I'm Wright. I'm a quantitative geneticist in the pre-breeding group here at NIAB. So my role is to look at the genetic makeup of the pre-breeding material that's been generated in a number of different projects. We do this by extracting DNA from the leaf tissue of that material and send away for genotyping. Typically we genotype on single nucleotide polymorphism arrays. So this is where we have thousands of DNA markers that form genetic landmarks across the genome. When this data is returned to us, we can combine it and compare how genetically distant potential donors are that we could use for bred wheat improvement, and also measure how genetically distant our pre-breeding lines are from existing varieties to ensure that we're working with something different. The genetic marker data is also important for being more accurate with the introgressions that we're making into a modern bred wheat background from a wild source. This is important because we want to avoid incorporating a lot of unwanted diversity from a wild background into that modern source, which is typically what you'd find in a wild background. So by using markers to facilitate the introgression of a certain chromosome region, we can be more accurate about only incorporating that region. So another part of my role is analysing the phenotype data that comes out of the field trials. This is important for removing field fertility effects from the phenotype data that we're actually interested in. So once we've done that, we can then use the genetic marker data that we generated and the field phenotype data, and we can use that data together in methods such as QTL mapping and genome-wide association to form marker trait associations. This is important because if we're trying to capture genetic diversity from a wild background, these marker and genotype associations can be used to facilitate marker-assisted selection and introgress that diversity from that wild background into modern bred wheat. So overall, we're combining the field phenotype data and the genetic data that we're generating to form pre-breeding lines with the aim of increasing genetic diversity within the modern wheat gene pool.